What's up guys, BIOBEAN here. So for the last little bit, we've been talking about evolution and how changes are inherited. So for the next little bit, we're going to be focusing on how we inherit the same genetic information from cell to cell. So let's get started. So there are two main ways that genetic information is passed from cell to cell. The first one is meiosis. Meiosis is the cellular process where your gametes are made. So that is your sex cells. So either your sperm, if you're a guy, or your eggs, if you're a female, those are made through the process of meiosis. And that gamete, that egg or that sperm is then going to meet either the egg or the sperm, and that will then create a fetus. So meiosis is done in order to create our gamete cells or our sex cells that will be able to aid in reproduction. And the second way that genetic information is passed on from cell to cell is the process of mitosis. Whereas the process of meiosis is generating gametes so that it can mix with someone else's cells to produce a new human being, the process of mitosis is essentially splitting one cell into two to create two identical copies of the same cell. So there's also a third way for genetic information to be passed on, but that is only found in prokaryotes, and that is called binary fission. Binary fission occurs in prokaryotes, which lack a nucleus, and so they have a little bit of a simpler time separating into two cells. Essentially, binary fission constitutes replicating the genome of a prokaryote and then splitting that genome off into two cells. But for the purposes of this lecture, we will be focusing mainly on mitosis, which happens in eukaryotic cells in order to generate an identical copy of the same cell. So let's get a little bit more into mitosis. Mitosis is the process where a eukaryotic cell whose genome has already been replicated separates into two separate cells that have genetically identical information. So mitosis is broken up into a few stages. And the way that I like to remember the name of the stages is using the mnemonic PMAT. Prophase, metaphase, anaphase, and telophase. So let's get a little bit into what happens at each stage. During prophase, we find that the nucleus starts to dissolve and the chromosomes start to kind of condense and get into that standard X structure that we think of when we think of a chromosome. And then the next stage, metaphase, is when the chromosomes all line up along the middle of a cell, getting ready to be pulled apart to either end. Then during anaphase, the chromosomes actually begin moving towards the edge of each cell, which will soon become two separate cells. And then during telophase, you get a contraction in the middle, which closes off each cell and creates two separate cells. So telophase is that phase where you finally separate into two cells. So this process of mitosis is involved within a greater process of the cell cycle. So let's get a little bit into what is the cell cycle. So while we just talked about mitosis, which includes the prophase, metaphase, anaphase, and telophase, if you've taken high school biology, you will also be familiar with the process of interphase. And generally that's considered the time in which the cell is doing all the normal cell-y stuff. If it's a kidney cell, it's filtering your blood. If it's a liver cell, it's also filtering your blood. If it's a heart cell, it's pumping your blood. So it's doing its normal function. And that interphase process occurs throughout this entire stage here. So interphase is coming from G1 through S to G2, and then eventually the cell enters into mitosis where it actually goes about splitting itself. So let's go over the different stages that occur within interphase. So the first stage that occurs in interphase is G1. So G1 is the time in which the cell is deciding that maybe it may want to start dividing. So it starts to grow bigger, it starts accumulating more size, more nutrients, and just starts to prepare kind of like a person would prepare for winter or a bear would prepare for winter. It's getting all of the things that it needs ready. And then there is the S phase. So that is the synthesis phase. So that is the time in which the genome is actually going to be replicated. So there's special proteins that go about actually replicating the genome and creating two copies of each chromosome. Then the G2 stage happens. So again, that's just the normal growing stage, they're replicating the organelles, they're just making sure that the cell has everything it needs for it to split into two separate cells. And then we enter into that mitosis phase where the cell actually splits its chromosomes and creates two cells. But there's also this little G0 phase here that's kind of external to the cell cycle. And the reason that it's kind of external to the cell cycle is because cells enter G0 when they're not actively cycling. So all of these processes, G1, S, G2, and mitosis, all of those are the cell preparing to split, synthesizing more chromosomes so that it can split itself, and then actually doing mitosis where the cells split. But G0 is the point in which the cells just live in and do their normal cell function. So again, if it's a liver cell, it's doing liver things. If it's a heart cell, it's doing heart things. And it's not really concerning itself about splitting. And most cells spend most of their time in this stage, right? Because if our cells were always splitting and not doing the actual function, you would die because your liver would be busy making more livers and not doing what a liver does. But there are actually some cells that stay in G0 their entire life. So if we think about, if you ever heard, don't hit your head or lose brain cells because you need all that you got. The reason for that is because brain cells or neurons actually do not enter into mitosis. So they live continuously in this G0 phase. So then that kind of begs the question is, why don't all cells live in the G0 stage? Why might we want some cells to enter into mitosis and grow? So let's get a little bit into that. So there's two main reasons why a cell might want to enter into the cell cycle and eventually create two identical copies of themselves. So the first one we can think of is when you grow, right? When you are a baby, you grow like feet within a few years. When you hit puberty, you hit massive growth spurts. And so your cells need to keep up, right? You're made up entirely of cells. So they need to continue to divide so there can be more of them to make up more of you. So if you think about a sperm that just hit an egg, those two cells that then merge eventually create a baby. And so you need an immense amount more cells than what you started with to create a full-grown baby. And then from a baby to a full-grown adult, all of those things requires your cells to divide and replicate themselves so that there can be more cells to make up more of you. And the second reason that we might need cells to enter the cell cycle is because we need tissues to maintain and repair themselves. If you've ever heard that you lose all of your skin within the span of a year, then you've heard of the idea of maintaining tissues. So our skin is constantly falling off because our skin is made up of many layers and the upper layer always falls off. You have injuries to your skin as well as inside you, like your intestines. If you don't chew your food a little too well, you feel that scraping down your esophagus. That is something that's going to need to be repaired and maintained. If you ever bite your cheek and now you're bleeding, that tissue needs to repair. Your heart tissue over a period of time starts to get old and it needs to maintain itself and create new cells. So that's the main second reason why we would want cells to enter the cell cycle and undergo mitosis, is because we need to maintain those tissues that exist in our body. So there are even cases where we want our cells to cycle rapidly. And we can think of that as when we get like a big cut on our arm. We would want our cells to very quickly close that wound as quick as possible. And so that's when we want as many cells as we can to be entering into the cell cycle and dividing so we can fill that gap that was created by a wound. But there are also some reasons why we might want our cells to actually kill themselves. And so that process is called apoptosis. So the process of cells killing themselves is called apoptosis. And that's when cells die in a programmed way. So why would cells want to die? Generally, cells have a lifespan. So as we said, any time that a genome is being replicated, there are going to be mutations that accumulate because that happens as a consequence of any genome that is going to be replicated. And so those mutations, if continue to allow to accumulate, can eventually start to harm the function of the cell. And now the liver cells aren't really doing what liver cells should. There's a little cluster of them, and they're starting to not do what liver cells do. Maybe they're starting to enter into mitosis when they shouldn't really be. We don't really need more liver cells, but they're growing regardless because they've accumulated so many mutations that they now no longer respond to the normal controls that a cell would. So that's when a cell will actually decide to undergo apoptosis. So the cell is going to tell itself that you're going haywire, you need to stop. And sometimes surrounding cells can send signals to that bad cell and tell it, hey, you gotta stop. And so they actually then undergo that programmed cell death, which is called apoptosis. And a really big reason why we need apoptosis is to prevent against cancer. So not only is it bad for a cell to stop doing what its normal function should be, because now that tissue is now no longer going to have as many cells as it should. So now your liver cells are functioning with only 75% of the cells that it would need in order to actually properly filter out your blood. So it's very important that cells do their proper function for that reason, but it's also very important because it helps protect against cancer. So cancer is essentially just the process of your normal body cells accumulating so many mutations that decides it doesn't want to be a liver cell anymore. It wants to just grow. That's just its whole desire, is to just grow and make more of it and more and accumulate a tumor the size of a melon. That's its whole goal. And so this process of apoptosis helps stop cancer before it starts. So when a cell starts to go beyond its normal controls, a cell will then decide to do apoptosis so that it will not become cancerous. Obviously, this process isn't perfect, and cancer cells have many ways to evade the normal responses of a cell, but it does a pretty good job because we're not all walking around with cancer, hopefully. Because it's so important to maintain cells in their normal state, there's also many checkpoints during the process of mitosis and the cell cycle to ensure that the cells are properly where they should be, everything is where it should be for them to make a new cell. Because you don't want to make a copy of an already bad cell, because that's a pretty easy way to get cancer to start to spread, is if you have no control or checkpoints in the way that they divide. So let's get a little bit into what the checkpoints are within the cell cycle. So there's a few points within the cell cycle that act as checkpoints to ensure that the cell is where it's supposed to be, everything is where it's supposed to be, in order to make sure that we don't get two bad cells. So we want to make sure that the same exact information is passed from one cell to the next. So the first checkpoint that we have is at the end of G1. So G1 was that stage where the cell is growing and accumulating nutrients in order to get ready to split that one cell into two cells. So there's a checkpoint at the end of that G1 phase to make sure, all right guys, are we ready? Do we have all the nutrients we need? Are we the size that we need? Do we have are the organelles ready? And if it says no, then it says, all right, go back into G1, continue to grow, continue to do what it needs to do. Or if it's so not ready, maybe it'll actually tell it, you know what, you're just not ever going to be ready. You should really undergo apoptosis and you should die. So that's always an option at any of these checkpoints is, you know what, you did such a bad job with that stage that actually you should just do apoptosis. You should do cell death. So the second stage that we see where there is a checkpoint to make sure that everything is happening correctly is at the end of synthesis phase, or at the end of S phase. So that's that stage in which the chromosomes are being replicated, getting ready to split into two separate cells. So it's super important that the exact same genetic information is passed from one cell to the next, because otherwise we might have our heart cell no longer doing heart cell functions, and that would be really bad for your heart. There is a checkpoint to make sure that the chromosomes were copied exactly the way they should be without any mistakes. Obviously, some errors make it through. It is not a 100% perfect process, nothing in nature is, but it does a really good job. And then there is our third checkpoint that happens partway through mitosis, and it happens at that metaphase step. So that is where the chromosomes are supposed to be lined up along the middle of the cell. So if a chromosome is oriented wrong, or maybe they're all bunched up, or they're lining up not quite in the middle, so there's going to be a checkpoint there that goes, stop guys, you're not correctly lined up. We need you to get you where you need to be. You need to be in the middle in order for you to do the rest of mitosis, so that anaphase and telophase stages. So if it passes that checkpoint at the metaphase step, then it is able to pull apart those chromosomes, and if not, then perhaps the cell might either fix whatever the error is or undergo apoptosis. So those are the 3 checkpoints within the cell cycle that are there to really ensure that the exact genetic information is passed on from cell to cell, and to make sure that the cell is correct. It's not going to have any issues. It's not going to be missing any organelles. It's not going to be super, super tiny. It's going to be exactly what it needs to be. So we just got a little bit into how a cell replicates itself, but let's get a little bit into what is it replicating. This is a little bit definition heavy, so bear with me. When we refer to a genome, we are referring to the total amount of DNA present within a cell. So that not only includes the DNA that is contained within the nucleus, but also in the mitochondria. So interestingly enough, the mitochondria also contains its own genome. And so that is, if you're an animal cell, your genome is referring to your nuclear DNA as well as your mitochondrial DNA. But if you're a plant cell, not only do you have nuclear DNA and mitochondrial DNA, but you also have DNA contained within your chloroplast. And the reason that we believe that the mitochondria and the chloroplast contain their own sets of genomes, or rather their own sets of DNA, is because we believe that they originated from a cell. So it's believed that many, many millions of years ago, a single cell ate another cell. And now the cell that got eaten is now either the mitochondria or the chloroplast. And we believe that it came from a bacteria because the DNA contained within the mitochondria and the chloroplast are actually circular. And so that's generally found within prokaryotic cells, is when the DNA is circular, whereas our nuclear DNA within eukaryotes is linear, right? We have those linear pieces of chromosomes. The next definition I want to introduce is the concept of N, or N value. What N value is the number of unique chromosomes that you have within the nucleus. So while the genome refers to how much total DNA that you have in your nucleus, in your mitochondria, and if you're a plant within your chloroplast, the N value refers to how many unique sets of chromosomes you have within your nucleus. We have 26 sets of unique chromosomes, but that's not the whole story, because we have 2 copies of each chromosome. One you inherit from your mother and one you inherit from your father. And so we refer to that as the coefficient of N. So how many sets of each chromosome do you have? In humans, our N coefficient, or the coefficient of N, is 2. So in humans, we would refer to our nuclear genome as being 2N. So the last concept that I want to introduce is C value. So C value is the amount of DNA in one set of your nuclear chromosomes. That's actually measured with a weight measurement. So generally it's expressed either in picograms, which is like even smaller than a nanogram, or expressed in base pairs. So how many base pairings are there? Humans have about 3.2 billion base pairs in one set of our chromosomes. But we have 2 copies of our nuclear chromosomes. So we refer to the C value in humans as 2C because we have 2 copies. So because we have the 2 copies, we add that coefficient to the C. So humans exist as a 2N and 2C genome. Humans have 2 copies of each nuclear chromosome, but they actually replicate themselves throughout the cell cycle. And so let's track where the coefficient of N and the C value is at the different stages of the cell cycle. So here's the cell cycle, and we're going to be going over what the N and C values are at the different stages. So I've left off G0 just for ease of completing this exercise. So in the G1 phase, the cells are at 2N and 2C. So their normal resting state for the human cell is 2N and 2C. Then we enter into the S phase. By the end of S phase, our cells have replicated the genome, and so we have shifted from 2N and 2C from G1 phase into 2N and 4C, because we now have double the amount of chromosomes, so double the amount of DNA, so double that C value. So we've moved from 2C to 4C. By the end of mitosis, the chromosomes that were replicated have now split into two separate cells, and so we return back to our normal steady state for a human cell, which is at 2N and 2C. So you may be thinking to yourself, well, that's odd. We doubled the number of chromosomes, but the coefficient of N remained the same, despite the coefficient of C doubling, going from 2C to 4C. But throughout that whole cell cycle, the coefficient of N was always 2. So why is that? In a normal cell that's sitting in G1 phase, there you have two sets of each chromosome, one that you inherit from your mother and one you inherit from your father. So don't let the strand confuse you. These are still the standard double helix that you would expect to see. We're just showing it as a single strand for ease of the explanation. So the way that we count unique sets of chromosomes is based on their centromeres. So centromeres are that middle portion of a chromosome. So in this stage, for this specific set of chromosomes, I count that there are 2 different centromeres. So that is why the coefficient of N was 2. But when we actually undergo replication, let's count the number of centromeres. When a chromosome undergoes replication, it actually sticks to the same centromere. So there's the same centromere going through now the copied chromosomes. And so while we have now doubled the amount of DNA we have, so we've now moved from 2C into 4C, we still only have 2 centromeres. And so the value of N continues to be 2. So anytime that you're unsure of what the coefficient of N should be, think about how many centromeres are there. So I mentioned that the total amount of human chromosomes within a single set of chromosomes is 3.2 billion base pairs. That seems like a lot of DNA. And so you might think that the more DNA you have, the more complex you are. So that would likely tell you that humans would have the most amount of DNA. But that actually does not hold to be true. And we actually find there is this C value paradox, where you would think that logically, the more DNA you have, the more complex you are. But we do not find that to be the case in nature. There are actually many organisms that have more DNA than we have that we would not consider to be more complex than humans. So we refer to this phenomenon as the C value paradox. So this concept that just because you have more amount of DNA does not mean that you are going to be a more complex human, or rather organism. So at this point, you probably have a bunch of C words swirling around in your head, so I'm going to try and clarify some of those. So you may have heard of this concept of a chromatid. So let me explain what the difference is between a chromatid and a chromosome. So here we have a common structure that people generally refer to in their brain when they think of a chromosome. And so I referred to previously that we count the number of chromosomes based on the number of centromeres. So here we have only 1 centromere, so we only have 1 chromosome. But that 1 chromosome is actually made up of 2 chromatids. So each half of this chromosome, it actually consists of 2 individual chromatids. So on one half, we have a first chromatid, and on the other side, we have a second chromatid. And they together make up a chromosome. And this is what a replicated chromosome would look like. And because it only has a single centromere, we refer to it as 1 chromosome. These chromatids are referred to as sister chromatids because they are identical copies of each other. So you can maybe think of them as identical sister chromatids. And so when you enter anaphase of the cell cycle, these 2 chromatids are actually going to be pulled apart into the separate sides of the cell. And then once they become separated, they're now referred to as their own individual chromosomes. So once the cell pulls the 2 chromatids apart, they now have their own centrosomes. So they are now referred to as daughter chromosomes. So from that original chromosome that was composed of our 2 sister chromatids, they then separate and form our daughter chromosomes. So now they're going to divide into their own cells, and now it is a chromosome in one cell and a chromosome in another. So that was how two sister chromatids split apart to become two daughter chromosomes. And that was for only a single copy of a chromosome. But I told you that our cells have two sets of each chromosome. So what are those called? So here we have a copy of, let's call it chromosome 1. And one of these is from your mother and one of these is from your father. So what we will see here would be following S phase after each of those chromosomes have been replicated. So now they consist of two pairs of sister chromatids. So each one has two sister chromatids. And we can count that each one has its own centromere. So what do we refer to as these two sets of chromosomes that carry the same type of information? We refer to these as homologous chromosomes. So when we have both copies of our chromosomes from our mother and father, we refer to them together as a pair of homologous chromosomes. Because they're not identical chromosomes, because while they contain the same type of information, let's say for instance, chromosome 1 carries the information for hair color. So both of these are going to carry the information from hair color, but maybe your father has black hair and your mother has blonde hair. And so that's going to be the slightly different type of information that's carried from one chromosome to the next. And so they are called homologous chromosomes. They are very similar chromosomes that carry the same type of information. So how do we know all of this about chromosomes and chromatids? So we've actually been able, for a while now, to look at the individual chromosomes. And it's easiest to do this when the chromosomes are super condensed, because when they're all spaced out in the nucleus during interphase, it's generally really hard for us to pick out each individual chromosome. So we want to capture the chromosomes when they are in their most tightest condensed form and when they're most organized. So that happens during metaphase of the cell cycle, or rather of mitosis. And so we have actually been able to take images of each individual chromosome. And then we can conglomerate them into one main image, and we call that a karyotype. So using a karyotype, you are able to look at how many chromosomes a organism has. So if you see multiple copies within each chromosome set, that you know what the coefficient of n is. So in the case of a human, we have 2 copies of each chromosome, and therefore our coefficient of n is 2. Something that can be a bit confusing when trying to read a karyotype is determining how many sets of chromosomes there are. And so because we're looking at a magnified view of chromosomes, they generally tend to look like only a single strand. But because we're in metaphase, we know that each chromosome has been copied, and each chromosome consists of 2 sets of sister chromatids. So even though in the karyotype, each chromosome set just looks like a single strand, we know that it is actually composed of 2 sets of sister chromatids. So don't get tripped up when you're looking at a karyotype, and make sure that you understand that when you see a single strand within a karyotype, that actually consists of 2 pairs of sister chromatids. So that concludes lecture 4. Stay tuned for more information about DNA and how it's replicated. And if you have any issues or any confusion surrounding any of the ideas presented here, please visit biologybean.com, where you can access all of your tutoring needs. If you have any questions that are very pressing, please feel free to mention them in the comments section, and I'm happy to answer them. Don't forget to like, share, and subscribe,